Good morning, students. Yes, in the last class, we were studying about the endosperm tissue. Yes, hopefully all of you remember. And also we studied about the artificial hybridization and also about the pollen pistil interaction. In the last class, I have given you a home assignment that you have to do and submit, write and submit me the artificial hybridization process. Only few of you have submitted, so I want all of you to submit it to me. Is it clear? Okay. Secondly, I also want you to revise these topics regularly. One of the important chapter this is, so I want you to revise it regularly, study it regularly so that you know the connection between it and you do not get confused. It is a very nicely one organized chapter it is, where from the start, that is from the pre-fertilization, fertilization and post-fertilization events are very nicely, they have been like clubbed in. Yes, very nicely they have been clubbed in. So you have to like regularly revise it so that you are aware. Okay, you are aware, and also you are being like, you're regularly understanding the topics. Okay, regularly understanding the topics, and you know that okay, what is happening, why it has been like connected to this, how endosperm is formed, how endosperm is connected to the double fertilization and all. So this is very important that you regularly study it. Okay, so I want you to also note down these points. Now, when you note down these points, what happens is when you're regularly noting down the points, so one thing is sure that you're regularly revising it. And by the end of the chapter, when I will give you the when I will send you the notes of it, so that you can like easily follow it up. Yes, easily follow it up. Regularly read your textbook as well. Yes, regularly read your textbook as well. And this chapter also contains a lot of very important neat questions. Yes, time and again I've been telling you, okay, this question can be asked, pollination can be asked, fertilization can be asked, about endosperm questions are asked. So there are many such questions which are included from this chapter as well. So that I want you people and every day to revise it regularly. Is it clear? Yes. And please submit your home assignments to me. Okay. So coming to this topic, that is endosperm, which we have already studied. It is a nutritive tissue formed from the primary endosperm nucleus that nourishes the developing embryo. Yes, there are two types of endosperm development. One is free nuclear type, one is cellular type. Okay, by the division of the cells, one of the endosperm formation, one by the free nuclear type. That is by the division of the nucleus. So coconut water from tender coconut, which is from the process of the nuclei division, and the white kernel which is formed in the coconut, you can see here. Yes, you can see here. So this is nothing but it is an endosperm, which is a nourishing tissue. Yes, it is an endosperm which is a nourishing tissue, and it develops from the primary endosperm nucleus. It develops from the primary endosperm nucleus. Okay, so this is about the double fertilization, students. You all know what happens here. When the pollen tube will enter into the synergids from the filiform apparatus, it will the male gamete will move. One male gamete will fertilize the egg, another male gamete will fertilize the polar nuclei present in the central cell, and this forms the triploid primary endosperm nucleus, and that triploid primary endosperm nucleus forms into an endosperm tissue. And what is that endosperm tissue? Endosperm tissue is nothing but the nourishing tissue which nourishes the developing embryo. The developing embryo. Okay. So coming to this embryo. Now, what is this embryo? Yes. So the definition is important. The definition can be asked. Okay. Definition is important. So the embryo is a sporophytic plant. Okay. The embryo is a young sporophytic plant present within a seed. Now, what do you mean by this? So, when we were studying in our first year, portions, you all know a sporophytic plant, how it is. A sporophytic plant which has got the leaves, the fruit, and the seta. Yes, the plant body. Now, in that small embryo, yes, in that small embryo, within the seed, present one small sporophytic plant. Yes, present a small sporophytic plant, which later germinates. Yes, so an embryo, what it is, a young sporophytic plant. The sporophytic plant is what? You know, it is divided into three: the fruit, seta, and the leaves of the plant body. Yes, so it is present within the seed. Inside the seed it is present. Where it is present? Inside the seed. So it is nothing but called as embryo. It is nothing but called as what? It is called as embryo. Okay, so you need to remember this definition. What is the definition? Very easy. One young sporophytic plant, the plant, you know, no, all of you know the plant, the parts of the plant, and also a young plant which is present inside the seed, which germinates later. Yes, which germinates later. Now, and there is also one process that is known as embryogeny. Now, what is this embryogeny? Embryogeny is nothing but early stages of development of embryo. Early stages of development of embryo is nothing but embryogeny. Okay, so first thing you need to remember is embryo. A young sporophytic plant present within the seed is nothing but called as embryo. Now, what is embryogeny? Embryogeny is nothing but the early stages of embryo development as the embryo develops into a plant, or in the human body, how it is the earlier embryo develops into fetus, that is the small infant baby. Yes, you know, no? Small, when you see any sonographic picture of a human womb, yes, human womb, if you have seen a small dot like structure, you see later as it goes from the trimester, from 3 months to 6 months to 6 months to 9 months. So what happens? There is the development of the embryo. Yes, there is a development of a fetus, a baby. Yes or no? It's a similar way. Here also, the young sporophytic plant present within the seed, embryogeny is nothing but early stages of embryo development. The early stages, the starting stages or the initiation stages of the embryo development is nothing but called as embryogeny. Okay. Now, next is zygote develops into proembryo and later into mature embryo, that is heart-shaped, this way. Okay, the heart-shaped. First, the zygote. Now remember this. What happens in this embryogeny? Stages of the embryo development. The zygote divides, forms a globular embryo. Yes, forms a globular embryo. Later, the heart-shaped embryo and into one mature embryo. Yes, zygote divides, forms a globular embryo, first into the proembryo, globular embryo, heart-shaped embryo, then the mature embryo. Yes, it is also as similar as a heart shape. So into a mature embryo. So these are the stages in embryo development in a dicot. Okay, stages in embryo development in a dicot, means dicotyledonous plant. So first, zygote develops into proembryo and later into mature embryo, that is nothing but called as embryogenesis or embryogeny. First, you see here, zygote is there, forms into proembryo, globular embryo. Here you can see here the cells are divided. Here only two cells are there. There are so many cells. Later it forms a heart shaped. Then there are divided into even more cells. There are suspensor, which has got there is a many cells here in the suspensor also. These are called a suspensor cell. And later into a mature embryo it develops. So you need to remember this. Clear? Now once again I will tell you what. First remember the picture. See students, anything, You need to either remember it in the flow chart or either remember into picture, at least biology. So what happens here? Now when I'm telling you, so I will tell you what. Okay, so this is a small zygote. If you remember this in the image wise or in the picture wise, so you can easily write it, yes or no. First, what happens? Zygote, then the pro-embryo, then there is the globular embryo, then there is a heart-shaped embryo, and then it becomes mature. Now, once you remember the picture in your head, once you remember the picture in your head, so that you can easily write it, yes or no, everyone can write it. Only you need to remember is the names. Okay, only you need to remember is the names. Once you remember the names, you can easily write it down, yes or no. First, what happens? You need to write it down in the point wise. Yes, so this is about the stages in embryo development. Now, the embryo consists of what all things? What all things the embryo consists of? Embryo consists of embryonal axis, cotyledons, plumule, and radicle. Yes, embryo consists of embryonal axis, cotyledons, that is the layer or the covering, plumule, and radicle. Okay, plumule and radicle. Now, this is the diagram of dicot embryo, and it is very important. It can be asked for you. Draw the diagram of dicot embryo and label the parts. Important question. Or the question can be asked in this way also. Draw the dicot embryo and explain the process of the formation of dicot embryo. Different stages of embryo development in dicots. So this can be asked as well. So you need to remember this is an important diagram, and labeling is even more important. Yes, unlabeled diagram do not contain any marks. No one will give you marks for unlabeled diagrams. So once again, I'm repeating. So there is a plumule. Yes, there is a plumule, cotyledons, which are two coverings. There is a hypocotyle, radicle, and a root cap. This part is the shoot part, and the radical part is the root part of the embryo. Root part of the embryo. Cotyledons, you know, coverings? Yes. So we will learn it more about the dicot embryo. It can be asked for you. Explain the dicot embryo. Explain the dicot embryo can be asked. Draw and explain. Now, what is this dicot embryo? Dicot embryo consists of an embryonal axis and two cotyledons. Yes, it consists of an embryonal axis and a two cotyledons. Dicot. Di means two, cotyledons means covering. Yes or no? So the how many cotyledons are present here? You see here, no? One cotyledon, another cotyledon. Two cotyledons are present, and so it is known as dicot. Okay. Now the portion of the embryonal axis above the level of cotyledon is called as epicotyle, which terminates with the plumule or a stem tip. Yes. So plumule is a stem tip here. Okay. Plumule is a stem tip. Yes. Which terminates with the plumule or the stem tip. So this part is known as the epicotyle part. Later, here is the hypocotyle, the cylindrical portion. Can you see here? This part is known as hypocotyle. Okay. Below the level of cotyledons, it is present below the level of cotyledon that terminates till the radical part. Yes, lower end in the radical or the root tip. The root tip is covered with a root cap. Yes, the root tip is covered. The radical part is covered with a root cap. So the cylindrical part is hypocotyle. Yes, the embryonal axis above the level of the cotyledon is nothing but the epicotyle, which terminates till the plumule, which is the stem tip, from where the stem or the shoot part arises. Where the stem or the shoot part arises, the cylindrical portion is nothing but called as hypocotyle, which terminates by the end of the radicle, which is covered with the root cap. Okay, which is covered with the root cap. So it consists of this embryonal axis and two cotyledons. Now, the portion above the embryonal axis of the embryonal axis above these two cotyledons is nothing but the epicotyle, which terminates, which stops by the plumule. The plumule is nothing but the stem tip. Okay. Then the cylindrical portion is nothing but is the hypocotyle. Yes, it is nothing but the hypocotyle, which terminates by the end of the radicle or the root tip. Okay, which is covered with a root cap. So this is nothing but the dicot embryo, which is nothing but the dicot embryo. And these are the points you need to give if it is asked for you. Explain about the dicot embryo. Yes, if it is asked for you, explain about the dicot embryo. So it is an important explanation is important, diagram is important. Okay, students. So next, coming to the different stages of embryo development in dicots. What are the different stages? Yes, what are the different stages of embryo development in dicots? So once again, I'm telling you. First, you need to know what is embryogenesis or embryogeny. Okay. Next is where the zygote is present. Yes. Next, how does it forms? This diagram is important. Once you understand the diagram, you can easily write the stages of embryo development or the embryogenesis. Okay. Now coming to the zygote is situated at the micropylar end. So all of you know micropylar end, where it is, where the micropyle is present. That is the small pore is present. Yes or no? The small pore is present. That is nothing but the micropylar end. The embryo sac divides and develops into an embryo. Embryo sac, you remember? Yes. The embryo sac consists of the antipodal cells, egg apparatus, then the central cell with the two polar nuclei. That embryo sac divides and develops into embryo. Okay, divides and develop into embryo. Now, where the zygote is situated? Zygote is situated at the micropylar end. Zygote, how it is formed? Zygote is formed from the fertilization process. From the fertilization, when the zygote is formed, zygote develops into embryo. Yes, zygote develops into embryo. That process from where the zygote it develops into embryo, that is nothing but called as embryogenesis. Yes, from the zygote the embryo is developed. Okay, from zygote the embryo is developed. So what happens here? The embryo sac which is present, yes, that oval shaped embryo sac which is present, it divides and develops into embryo. Now, the early stages of development of embryo is called as embryogeny. The early stages of the development of the embryo is called as embryogeny. From the zygote, as it divides and forms into embryo, that process is known as embryogenesis. That process is known as embryogenesis. So you need to remember, students, first, different stages of embryo development in dicots. So this is what you need to remember. So these are the different stages of the embryo development. Clear? These are the different stages of the embryo development. Most zygotes divide only after some amount of endosperm is formed. Yes, because why? Only some zygote will divide only after some amount of endosperm is formed, because endosperm is a nourishing tissue for the embryo. Yes, it nourishes the embryo. It is the nourishing tissue. It nourishes the embryo. It provides the nutrition required for the embryo development. So only once the endosperm is formed, the zygote will divide into embryo. Okay. So now what will happen first? See here, students. So this part I will tell you, explain it to you here. So the zygote undergoes mitotic division. Mitosis, you know, equational division. When the zygote undergoes equational division, it will form two cells. How many cells? Two cells, namely basal cell and apical cell or the terminal cell. Yes, the basal cell and the apical cell, that is by the mitotic division. Listen to me carefully. What happens here? We are now studying about the different stages of embryo development in zygotes. And the whole process is known as embryogenesis. And who develops into embryo? Zygote develops into embryo. Zygote develops into embryo. Now what happens here? The zygote undergoes mitosis. What it undergoes? It undergoes mitosis, and after undergoing mitosis, it forms two cells. How many cells? Two cells. One is basal cell, one is apical cell. Later, the basal cell divides further. Yes, it divides further, forming 6 to 10 cell structure known as suspensor. 6 to 10 cell structure known as suspensor. Can you see here? The basal cell will form into the suspensor cell. The basal cell will divide into the suspensor cells. Clear? Now, the other terminal cell divides continuously. The terminal cell which is there, it divides continuously to form a globular embryo which later becomes a heart-shaped. Yes, the terminal cell which is present here, two-cell embryo. Can you see here? The two-cell embryo. After once again, when the basal cell and the terminal cell undergoes cell division, basal cell will divide into 6 to 10 cell structure, whereas the terminal cell will divide into two-cell embryo. Two-cell embryo. Later what happens? The terminal cell will form into embryo. Okay, first four, then a globular structure, one round structure. And this 6 to 10 cell structure is nothing but called as suspensor. It is called as suspensor. Yes, so this is an embryo, this is a suspensor cell. Later what happens? It forms into a globular stage where the embryo is in the globular structure, one round mass-like structure, and this is the suspensor again. Later again it divides into heart-shaped embryo stage. What it is? Heart-shaped embryo stage, and this is the suspensor cells. These are the suspensor cells. And later it becomes into a mature embryo with two cotyledons, plumule and the radical part. Plumule, two cotyledons, and the radical, that is the shoot part. Yes, radical, which is the shoot part, which we call it as torpedo stage. That is the mature stage of the embryo. Clear? And this whole process is known as embryogenesis. So what happens here, students? Different stages of embryo development in dicots. Dicots means two cotyledons. What happens first? The zygote divides, and it divides only when there is a presence of endosperm tissue. Why? Because it nourishes the developing embryo. It nourishes the developing embryo. The zygote first divides, that is the mitotic division takes place. It forms into two cell. Yes, two cell. One is the apical cell, one basal cell. The apical cell divides into two cell embryo, whereas the basal cell will divide into 6 to 10 cell structure. Those are nothing but called as suspensor cells. Later what happens? The embryo will form into a globular structure, one round mass-like structure, and 6 to 10 suspensor cells are present. Later again what happens? The globular stage, from the globular stage, it forms into a heart-shaped embryo along with the suspensor cell, and later it divides further to form a mature embryo where two cotyledons are present, the plumule, hypocotyle, and the root or the radical part. Root cap and the radical part. So these are the different stages of embryo development. The process is known as embryogenesis, from the development from the zygote, where the zygote divides to form into embryo. where the zygote divides to form into embryo. So these are the different stages of the dicot embryo. Later, we will be studying about the monocot embryo as well. And before we move on to the monocot embryo, first let us just once revise what we have studied today. So about the embryo is nothing but a young sporophytic plant. Yes, it is a young sporophytic plant present within the seed. Yes, it is wrapped within a seed. Here, next is embryogeny. What do you mean by embryogeny? Embryogeny is nothing but early stages of embryo development. Early stages of embryo development where the embryo develops from the zygote. First, the zygote divides into two cell structure, that is the proembryo, terminal and the basal cell, and later divides into mature embryo. That is a heart shape. Embryo consists of embryonal axis, cotyledon, plumule, and radicle. You got to know in the embryonal axis, there is an epicotyle. Below that, there is a cotyledons. Then the plumule, which is a shoot tip. There is a hypocotyle, which is a cylindrical mass-like structure, and the radicle, which is a root tip. So this diagram is important. Later, the dicotyledonous embryo diagram is important, consisting of plumule, cotyledon, hypocotyle, radicle, and the root cap. Yes. Later, about the dicot embryo, you need to give the diagram plus explain it. Explaining about the embryonal axis, where the embryonal axis cotyledons are present, what the cylindrical portion, where what it is called as hypocotyle. There is a radicle and a root cap. Radicle is a root tip, plumule is a shoot tip. So this is, you need to remember about the dicot embryo. Later, coming to the different stages of embryo development in dicots. So different stages means the different stages where zygote develops into two cell structure, that is terminal cell, basal cell. Later, it divides into embryo, then the suspensor cell, which it goes from the globular stage to heart-shaped stage and the mature embryo. So this explanation is important. Plus, what is embryogenesis and how zygote divides. Zygote divides only when there is a presence of the nourishment tissue, which is the endosperm. So we studied about the dicot embryo today. If there is any doubt regarding this, so you can personally ask me or message me about the same. If you have not understood any topic, or if you want me to repeat it once again, you please can inform it to me. Clear? Secondly, please do write down and take all the important points, the diagram. The questions are time and again asked for you in your question papers as well as in your NEET question papers. So please make sure you revise regularly. And have a nice day, students.